Hello everyone. Today we will be discussing about the prokaryotic and eukaryotic cells structure and function, which is under the section of microbiology. And this question came in the year 2019 and the question number was 13. The question states that the granules are inclusion bodies found in bacteria. Which of the following statements are true about granules? Granules can be storage vessels for carbohydrates or lipids. Some granules supply substances for a variety of metabolic processes. Granules are dissolved in cytoplasm. Granules are membrane enclosed structures. Okay, so they have given you four options about something called as granules in or also some inclusion which are also known as inclusion bodies in bacteria, and they have asked you to select what are the correct statements or what are the true statements. Okay, so for solving this question, first you need to know what are storage granules or granules of inclusion bodies in bacteria. So first let me discuss about that. So if you see, this is an image of a schematic diagram of a bacteria where you can see all the general parts which a bacteria comprises. Okay, so, so bacteria is a prokaryote, so it does not have any nucleus. It is anucleus in nature. So it has a cytoplasm in which there is a nucleoid structure. Normally, the DNA is wrapped around in such a way that it is called as a nucleoid structure, and it has pilus and fimbriae for its locomotion, and the pilus also helps in its conjugation process. And what is important that I want to draw your attention in this slide is about the storage granules. So there are certain red dots that you can see in this picture which are known as the storage granules. So what are the storage granules? Storage granules are membrane bounded vesicles containing condensed materials. So this is either membrane bounded or may not be membrane bounded. In some of the storage granules, they are membrane bounded that I will be discussing, but it is not like that all of the storage granules are membrane bounded. Okay, but there are certain vesicles which actually contain, they are certain containers of nutrients or something essential which can be utilized by the bacteria in the stage of starvation, which will actually help the bacteria to survive that harsh conditions. So that is a very interesting thing that this bacteria possess. So these granules are like certain magic containers from where you can get different supplements that can actually help them to survive any stressful conditions. Okay, so they are also known as zymogen granules or condensing vacuoles. Storage granules are an important component of metabolism in many organisms spanning the bacterial, eukaryotes, and archaeal domains. These granules are the parts of the cell that store the cell's energy reserves as well as other important metabolites. So it is a reservoir of energy storage as well as certain important metabolites that is actually driving the cell to survive in very harsh conditions. Okay, and also there are the what are the functions and examples of this kind of granules? Granules found in plastids or in the cytoplasm assumed to be food reservoirs, often of glycogen or other carbohydrate polymers. So it can contain carbohydrate or glycogens stored in it. Okay, in prokaryotes, nutrients reserves may be stored in the cytoplasm in the form of glycogen, lipids, polyphosphate, or in some cases sulfur or nitrogen. Sulfur granules are essentially common in bacteria that use hydrogen sulfide as an electron source. The lysosomes of plant cells are membrane-bound storage granules containing hydrolytic digestive enzymes. Example: large vacuoles of parenchymatous cells of corn seedlings, protein or aleuron bodies, and starch granules of cereal and other seeds. So it may also contain certain lysosomes, for example, which can actually help in which has a hydrolytic digestive activity for various components which can actually help this bacteria or the prokaryotes. So this I am talking about the plant cells. So there it can actually help the plant cells to combat any foreign pathogens, okay, which can actually infect that plants. Okay, so there these lysozymes can be also very important. Okay, now, so what are inclusion bodies? So these storage granules are sometimes also called as inclusion bodies in the bacterial cells. So now inclusion bodies has various definitions if you search in the net. So someone will tell you that it is a aggregate of a lot of proteins. Okay, and also, but the major sense of the inclusion body is same, that it has a this has these are the elementary bodies that are cytoplasmic inclusions or nuclear aggregates of stable substances like proteins. So they are actually aggregates, or I can say that they are actually a storehouse of many materials together, which are actually stable substances like proteins or other metabolites. Okay, and they are present in an intact form in the cytoplasm. Okay, so they are actually floating in the cytoplasm of the bacteria. They freely suspend and float within the cytoplasmic matrix and can also be called cytoplasmic inclusions. Some inclusion bodies lie free in the cytoplasm, whereas some are enclosed by a shell. So that is what I was telling in the first sentence, that storage granules are always membrane bound. It is not the case. Sometimes it may be also floating like in free in the cytoplasm, or sometimes it can be enclosed by a shell. Okay, so they are usually sites of viral multiplication, thus in a bacteria or eukaryotic cell, therefore are also referred to as viral inclusion bodies that consist of viral capsid proteins. So these inclusion bodies are also targeted by certain viruses, and they are the main region of the viral multiplications. So these inclusion bodies are also used to test that whether this how much virus have been in are infecting that bacteria or that prokaryote. Okay, and these are that's why they are also called as means viral inclusion bodies. Actually, that they contain these viral capsid proteins in it, which will be important for the virus to generate a complete virion particle. Okay, so this is about the inclusion bodies. So if there are two types of inclusion bodies, one is the organic inclusion bodies and another one is the inorganic inclusion bodies. Normally those falls under these organic inclusion bodies, which actually store certain organic compounds inside it. Okay, and those which are actually stored in some inorganic substances like sulfur, nitrogen. So those are coming under these inorganic inclusion bodies. So in organic inclusion bodies, you have glycogen granules, poly B hydroxybutyrate granules, and metachromatic inclusions, which actually contain this starch and glycogen. Okay, then there is a special kind of bacteria which can actually independently perform its photosynthesis, and they are the cyanobacteria, and they actually contain large polypeptides, normally containing arginine and aspartic acid. One which is actually which is acidic amino acid, and another one is a basic amino acid, and they these together forming large polypeptides, and they are called as the cyanophysin granules. Okay, and they also contain something known as carboxysomes. So these carboxysomes are containing certain pins. They are actually present in those bacteria which actually are looking for carbon fixation. Okay, so these inclusions contain the enzyme ribulose 1,5 diphosphate carboxylase, and also they are also the source of nitrogen. Okay, then gas vacuoles. This gas vacuoles is very interesting because that actually helps the aquatic prokaryotes or aquatic bacteria which are present in the deep sea levels, and they actually helps them to provide them a buoyancy, or they help them to actually float in the depth of that means sea level. Okay, so that's why these gas vacuoles are very interesting, which are found in certain aquatic prokaryotes. Then there is also something known as magnetosomes, which actually some bacteria orient themselves within a magnetic field. So this is also very important. So some bacteria, they have the tendency, if you put a magnetic field, they have the tendency to orient themselves towards that magnetic field. And because of the presence of these magnetosomes, which contains this magnetite, which is a mineral ore, or a mineral for iron oxide, that is Fe3O4. Okay, and these magnetosomes are normally membrane bound, inclusion bodies. Okay, so that's why I said that some are membrane bound and some are not. So magnetosomes are membrane bound inclusion bodies. Okay, then there are also certain lipid inclusions which contain certain lipid moieties that will be actually helpful for the bacteria to survive certain, or the prokaryote to survive, especially in the condition where they are actually facing some stress. Okay, so now, after knowing the basics about inclusion bodies, so the main points that I want to point out is that inclusion bodies are present in the cytoplasm and they are floating in the cytoplasm, whereas they are important reservoirs for certain important metabolites of the prokaryotes, which they can utilize in certain harsh conditions. They may be or they may not be membrane bound, okay, but they are actually storehouse of certain important metabolites. Okay, by knowing these things, let us go and revisit the question. So they are asking that granules can be storage vessels for carbohydrates and lipids. So as I have mentioned, there are liposome. There are two kinds of. So there can be starch glycogen that can be stored in the inclusion bodies, and also there are lipid inclusion bodies. So both of them can be stored inside the inclusion bodies. So from that we know that the statement one is correct. Some granules supply substances for a variety of metabolic processes. So they are important metabolites. That's why we are actually the prokaryotes are actually storing those things in the inclusion bodies. So the second statement also holds true. The third one is the granules are dissolved in the cytoplasm. But what we know that granules are not dissolved in the cytoplasm, they are present, they are floating in the cytoplasm. Okay, so for that, this sentence is not correct. Then granules are membrane enclosed structures. So some can be membrane enclosed, but not all of them are membrane enclosed structures. Okay, so from this we know that the option 1 and the option 2 are correct. Okay, so now let us look into the solution. So I have written the same thing. There are numerous inclusion bodies or granules in the bacterial cytoplasm. These bodies are never enclosed by a membrane and serve as a storage vessel. Glycogen, which is a polymer of glucose, is stored as a reserve for carbohydrates and energy. As we have already seen here, glycogen and starch in metachromatic inclusion bodies, they stored this. And many bacteria possess lipid droplets that contain polymeric esters of poly-beta hydroxybutyric acid or related compounds, just like that. Poly-beta hydroxybutyrate, which are also coming under these lipid inclusions. Okay. And the last one is in bacteria, storage granules are produced under favorable growth conditions and are consumed after the nutrients have been depleted from the medium. Okay, so these are actually produced when they are in the favorable growth conditions, and they are actually consumed when these nutrients, when they are in a stress condition. When they require these nutrients, then only it gets consumed. But it doesn't mean that they get dissolved in the cytoplasm. So that statement was wrong. So the answer to the question will be option C. So what is your option C? That is option 1 and 2. Okay. So the correct answer will be your option C. For for your uh, for your reference, you can actually look into these two courses. So the first one is Advanced Microbiology by Arup Kumar Mitra of EMMRC Kolkata, and the second course is similarly Microbiology by Dr. Gurvinder Kaur Saini. So these are two NPTEL courses you can look into for knowing more about this microbiology and this section that I am talking about.